Dear students, today we are going to study about the Rickettsias, Chlamydias and Mycoplasmas. Let me recall for you that in the previous lecture we had studied the Burgess system of classification. The system involves classification of bacteria into 4 volumes. And if you could remember, we had 4 volumes, and the first volume contains 11 sections. The section number 9 contained Ricketsias and Chlamydias, and that section number 10 contained Mycoplasmas. So since these are very important, we should have some knowledge of Ricketsias, Chlamydias as well as of Mycoplasmas. Therefore, today we are going to study Ricketsias, Chlamydias and Mycoplasmas. These are the two, Ricketsias and Chlamydias are the two subgroups, and that volume contain gram-negative bacteria. They belong to first volume. The Ricketsias are pleomorphic, that means they could change their shapes frequently, and they are non-motile. Also, they do not form spores. They are gram-negative and obligate intracellular parasites. Now, what is this obligate? If an organism is obligate parasite, it is also called as holoparasite. That means it cannot complete its life cycle without exploiting an appropriate host. If it fails to obtain a host, it will fail to reproduce and will die. The Ricketsias, they live in parasite, or they create a mutualistic association with certain vertebrates and arthropods. Their wall contains a specific compound called as muramic acid. It has 3 genera: Bartonella, Grahamella and Rickettsia. These are the 3 genera, and they causes Q fever, and the Q fever is caused by Coxiella burnettii, and its vector is lice. Typhus is another disease caused by Rickettsia. The vectors are rats, fleas and lice. Rocky Mountain spotted fever is caused by Rickettsia rickettsii. It is transmitted by tick. That means the tick is the vector for this disease. Then we have Chlamydia. They are also called as energy parasites. They are also gram-negative, belong to the first volume, in which contains all the gram-negative bacteria. It is also non-motile, cocoid and obligate. Multiply within the membrane-bound vacuoles in the cytoplasm of human beings, mammals and birds. It depends on the host for energy, as they are not able to oxidize glucose, so cannot produce ATP. Hence, they utilize the ATPs produced by the host itself. Their body is extraordinarily permeable to ATP and acetyl coenzyme. For this reason, they are designated as energy parasites. The diseases caused by Chlamydia are trachoma, which is also called as conjunctivitis, which is the disease of eye, caused by Chlamydia trachomatis. Then venereal disease by lymphogranuloma venereum, it is also a chlamydia. Then come to the mycoplasmas. Mycoplasmas are pleomorphic. That means they could change their shape frequently owing to lacking of the cell wall. Hence, they are termed as biological jokers also. They also belong to Burgess' first volume, and the section number is 10th and are gram-negative. They form colonies which are often termed as fried egg colonies. They are in fact the wall-less bacteria, also called as molecules. Mycoplasma are prokaryotes devoid of cell wall. Since they cannot synthesize peptidoglycan, these animals, they are extremely resistant to penicillin, but they are sensitive to lysis by osmotic shock, detergents and alcohols. The reason is they don't have cell wall, and the function of the cell wall is to prevent the bacteria from osmotic shock it suffers. They are pleomorphic, as I've told you in the beginning, and of the size of 0.3 to 0.8 micrometer in diameter. They are facultative or obligate anaerobes. Facultative means they have options, and obligate means they don't have any option. They are so small that they can pass even through bacterial filters. That means their sizes are lesser than that of bacteria, but slightly more than that of viruses. They require sterols and fatty acids for their growth. They are the parasites, commensals, or saprophytes. Saprophyte, as you know, is the mode of nutrition in which the organism draws its nutrition from dead and decaying matter. The genus of Mycoplasma are Mycoplasma mycoides, Spiroplasma, the Spiroplasma citri. The Mycoplasmas, since they are bacteria, they can be cultured in non-living media, unlike viruses which cannot be cultured in non-living media. Their genetic material is naturally DNA and reproduces by budding, and they never reproduce by fission. They are insensitive to penicillin but are killed by tetracyclines. They also causes diseases in human beings. The Mycoplasma pneumoniae causes upper respiratory tract disease in human beings. Mycoplasma hominis and Mycoplasma fermentans causes infertility in man. Another prokaryote we have are the Archaebacterias. They don't have nucleus, and also they don't have peptidoglycan. Nucleus, since they don't have nucleus and the genome is laid outside the in the cytoplasm itself, that's why they belong to prokaryota. The Archaebacterians, they are methanogens that they respire anaerobically, and O2 is lethal for them. The halobacteria, they live in extremely salty environment. That means they are halophiles. In fact, they love salt too much and can thrive in the salt water. Otherwise, other bacteria, they got plasmolized in the salty environment. The third one are thermoacidophiles. They live in hot acidic environment, and they found in acidic sulfur springs and utilize sulfur as electron acceptor. So students, I have two slides to show you regarding Rickettsia and Chlamydia. This is the mountain spotted fever which is caused by Rickettsia, and it in this type of fever you can see in the slide that how red spots and rashes they got developed on the entire part of your body. So you can see this picture which is drawn from internet. This is these are the vectors of typhus fever, which is a disease caused by bacteria in the genus Rickettsia, and it is acquired through the bite of an infected arthropod. This is Xenopsilla, and this is flea, and these are ticks and mites. So this is all for this lecture. Thank you so much.